Hello, here I would like to try to correct some of the misconceptions regarding the topic of neoplasia. And this is something that we found out when looking at your answers during the recent continual assessment. So the first subtopic that I want to talk about is dysplasia. One of the common misconceptions is that dysplastic cells are infiltrative and have invaded the basement membrane. So this is actually incorrect. By definition, dysplasia is an intraepithelial process. So this means that the epithelial cells are still bound by the basement membrane. There is no invasion through the basement membrane or into the stroma. Another misconception is that dysplastic cells are poorly differentiated or undifferentiated. This is not so. Differentiation is a term that we usually use to describe invasive cancer, established cancer that's already gone through the basement membrane. We don't use it for dysplastic cells. In terms of differentiation, the dysplastic cells also do definitely resemble the tissue of origin. They're not totally different and unrecognizable. They're still usually quite readily recognizable. Another misconception is that dysplastic cells is not neoplastic. Well, this is not actually correct. Dysplasia is an early part of the neoplastic process. Dysplastic cells, some have written that if the cells show pavementing, this is a feature of dysplasia, but actually pavementing is simply a feature of squamous differentiation. Especially when you see in simple squamous epithelium, the cells are quite large and they sort of fit quite nicely into each other. So this indicates squamous differentiation, meaning that the tissue is of squamous cell type, the epithelium, and it does not mean that it is dysplastic or neoplastic. Even benign squamous epithelium shows pavementing. Another incorrect idea is that dysplasia is encapsulated. Actually, dysplasia is not mass forming at all. It is an intraepithelial process, so it involves the epithelium within the confines of the basement membrane. So grossly, we actually cannot see it. It's either normal, the organ would look normal grossly, or if we look at the mucosa, we may see a little bit of color change, but there is no mass lesion. If there is a mass, this would already be a tumor or a neoplasm, whether it is benign or malignant. So a few more points about dysplasia. It is part of the neoplastic process. The cells have some genetic changes, but this is still early in the course of malignant transformation, so they do not yet have the capability to invade the basement membrane or stroma and to metastasize. So it is known as pre-malignant change. We use this term interchangeably with intraepithelial neoplasia, meaning that it's still confined within the basement membrane. What tissues undergo dysplasia? Well, epithelial tissues. We generally don't talk about dysplasia in mesenchymal neoplastic conditions. So squamous epithelium, such as those that we see in the skin, in the uterine cervix, or metaplastic squamous epithelium in the lung. Glandular epithelium, such as tubular adenomas in the colon. For characteristics, the cells are still bound by basement membrane. There can be varying grades of dysplasia. We don't use the term well differentiated or poorly differentiated, but we do say low grade, moderate, or high grade. And the highest grade of dysplasia, the most severe instance, would be carcinoma in situ, generally appreciated as the one with the highest risk of progression to invasive carcinoma. In terms of morphology, we must remember that, again, grossly, we generally don't see anything. Certainly there is no mass. There may be a change in color because the mucosa may appear a little bit reddish or paler, but there is no mass lesion. Microscopically, we do see some similar features to malignant cells, such as increased NC ratios, irregular nuclear membranes, prominent nucleoli, increased mitosis, hyperchromasia, and so on and so forth. The most severe changes are seen in carcinoma in situ. So, for example, in a stratified squamous epithelium, we see full thickness dysplasia starting from the lowest part, which is closest to the basement membrane, the basal layers, going all the way up to the more superficial layers. Then we call it carcinoma in situ or high-grade dysplasia. Whereas in low-grade dysplasia, generally the changes are seen more readily in the basal layers. The cells do not breach the basement membrane, they do not invade the underlying stroma, and consequently there is no evidence of desmoplasia. So this is just to show you benign stratified squamous epithelium. Here we see the basal layer. You notice that the nuclei are sort of arranged a little bit more vertically here, whereas as they mature towards the superficial surface, the nuclei start to become more horizontally arranged or become more flattened. This is polarity, nuclear polarity, or maturation. Now, in contrast, in high-grade dysplasia, when you can see the basal cells are still also quite vertically arranged, but we do not really have that flattening towards the surface, so there is loss of nuclear polarity. And here we can see that the nuclei are larger, they're more irregular, some of them have got prominent nucleoli. There are also mitotic figures seen in the basal third, also even in the middle third, and perhaps the superficial third of the epithelium. And here we can see the prominent nucleoli. So these are the features seen in high-grade dysplasia. Please note that the basement membrane is intact. There is no invasion into the underlying stroma. Now, looking at the mechanism of HPV-induced cervical intraepithelial neoplasia, one common misconception is that the main mechanism is by insertional mutagenesis. So this is incorrect. The mechanism is actually by viral oncoproteins E6 and E7 acting on host proteins, which are the gene products of P53 and RB. So they don't actually cause mutations at the gene level, but more interact at the protein level. So again, in line with this, the misconception was that E6 and 7 genes act on P53 and RB genes, or E6 and 7 proteins act on P53 and RB genes. So again, remember that it is protein interacting with protein. E6 and 7 protein or gene product acting on P53 and RB proteins or gene products, not the genes themselves. So again, and the other inaccuracy was that a lot mentioned that P53 and RB genes are the ones that bring about the regulatory effects. And again, it's not the genes, it's the proteins or the gene products that have these effects on the cell cycle and DNA repair and apoptosis. Now, continuing on with the grading of cancer, there is a misconception that grading is used to decide if a neoplasm is benign or malignant. This is actually incorrect. Both grading and staging are performed already when we decide that a tumor is malignant. In other words, on cancer. Cancer means that by definition it is malignant, it is infiltrative, and it can spread via lymphatics to lymph nodes and blood to distant sites. So it is not correct to say that low-grade tumors are benign, whereas high-grade tumors are malignant. In cancers can be low-grade or high-grade, but they're already cancer. There's also a misconception that in low-grade or well-differentiated tumors, the cells look normal. Actually, in low-grade tumors or well-differentiated tumors, the cells are already cytologically malignant, so they do not look normal, they do not look benign, but the tumor cells and the architecture still resembles the benign counterpart. So for example, in an adenocarcinoma, in a malignant glandular tumor, you can still see gland formation, or in a squamous cell carcinoma, you can still see keratin pearl formation. There's also a misconception that in breast carcinoma that the myoepithelial cells are still present in low-grade carcinoma. Again, this is wrong. Myoepithelial cells are absent once it is an invasive carcinoma. They are only present in ductal carcinoma in situ and in benign proliferations. Moving on to continue on grading, there's a misconception that in lung carcinoma, low-grade carcinoma shows ciliated columnar cells, whereas in high-grade carcinoma we see squamous cell carcinoma. So again, this is incorrect. Each carcinoma type can be low or high grade. For example, adenocarcinoma can be low grade or well differentiated, or high grade or poorly differentiated. Similarly, squamous cell carcinoma can be low grade or high grade. These malignant tumors do not change types from adeno to squamous, from low to high grade. Also, we see ciliated columnar cells almost always in the context of benign epithelium. In adenocarcinoma, we do not see cilia. So let's move on to staging. One of the misconceptions is that grading is part of staging. So this is incorrect. Grading and staging are both different, independent ways to assess tumor behavior and prognosis. There's also a misconception that low-stage tumors are benign and encapsulated. So as I mentioned earlier, staging is applied to already established cancer. In other words, we already know that it is invasive, or at the very least carcinoma in situ. there is no benign encapsulated stage. The earlier stage is TIS, in situ carcinoma, which is included in most of the staging systems just for completeness sake. Most of the time the tumors are invasive, so we're looking at a score from T1 to T4, and these are of course tumors that have already penetrated the basement membrane, and we would look at parameters like tumor size, depth of penetration of the primary organ, and also involvement of adjacent tissues. So this is an example to show you how colorectal carcinoma is staged. T1, where there is invasion just into the submucosa. T2, where it is gone into the muscularis propria but not beyond it. T3, where it's gone through the muscularis propria into the subserosa but not breached the peritoneum, and then T4, where it has gone through the serosa, perhaps also involving adjacent structures. So this is the end of this short session, and I hope you have found it helpful. Thank you.